Hey everyone, today we are looking at the citric acid cycle, or sometimes called TCA or tricarboxylic acid cycle. The citric acid cycle is part of a larger process that we'll be going through called cellular respiration. Now, when looking at the pathway glycolysis, which we previously covered, you can see that only a small amount of energy relative is actually captured from a glucose molecule by going through the process of glycolysis. If we were to convert this into energy units, it would be about 146 kilojoules per mole. And that is the process of taking glucose to 2-pyruvate through glycolysis. Now, cellular respiration, the whole of cellular respiration, is the full oxidation of glucose. It produces it uses 6 oxygen molecules, and it produces 6 CO2 molecules and 6 water molecules. So again, this is the full breakdown of all carbons by oxidation of glucose. And you can see that it nets 2,840 kilojoules per mole, much, much, much more than just glycolysis alone. So cellular respiration is the main source of energy, the main pathway that is utilized using glucose and breaking down glucose. So again, the takeaways here is that full cellular respiration does consume oxygen and produce carbon dioxide. Thus why we as animals, and in cases of plants as well, if they're using cellular respiration, consume oxygen, right? We have to take in and breathe oxygen, and we breathe out carbon dioxide. This is all a grand process of our cellular respiration taking place in all of the cells at the same time, coherently in our self as an organism. This is also why, and when we talked about glycolysis, cellular respiration only can occur during aerobic conditions because it requires oxygen. Now, it does provide much more ATP from glucose than glycolysis. Remember, glycolysis only nets you 2 ATP. It also can be used to get energy from lipids and amino acids. So it is a pathway that can be utilized by other types of energy molecules, not just sugars like glucose. There are 3 major stages during this process of cellular respiration. The first stage is producing acetyl-CoA. The second whole stage is the oxidation of acetyl-CoA. And then the last stage is actually doing electron transfer and oxidative phosphorylation. We're going to be going through all of these stages, but let me just point out that the citric acid cycle, as we call it, is technically one of these stages, stage 2. But we're going to be talking about this portion, acetyl acid CoA production, the acetyl acid or acetyl CoA oxidation, which is the citric acid cycle. And lastly, in further chapters, we'll be talking about this whole process of electron transfer and oxidative phosphorylation. So here is a look at that first stage of respiration, which is called the acetyl-CoA production. Now remember, this can be fed into from various pathways. As I noted, amino acids can feed into respiration, fatty acids can feed into respiration, and what we'll be looking at in particular, pyruvate from glycolysis can feed into respiration. Now, these other pathways, such as from amino acids and fatty acids, these catabolic pathways that produce acetyl CoA, we will look at again in other chapters. There are lots of mechanisms and pathways that's happening in this process right here to take amino acids or fatty acids and convert them into acetyl CoA. Today, we'll be talking about how pyruvate, our product of glycolysis, is converted into acetyl CoA. Now, this stage of taking pyruvate to acetyl CoA is going to generate some ATP. It's going to generate some NADH. It also generates another molecule, FADH2, which is again a similar high energy compound similar to NADH, and we'll be talking more about that. And it's going to help, again, release some of the energy from the pyruvate complexes as CO2. A grand look at respiration stage 2, again, is what we call the citric acid cycle. You can see here. The acetyl CoA we made in the first step of stage 1 of respiration is then fed into the citric acid cycle. Here, we're going to generate even more NADH, more FADH2, and we also generate some GTP. Now, the remaining carbons that are left over from this process are going to be potentially cycled into this continuing through the cycle. As you can see, we enter in and exit with a molecule, but through the process lose some carbons to CO2, and we lose lots of electrons as we oxidize to these electron-carrying molecules. All of the electron-carrying molecules that we've generated throughout the process of respiration and even through other processes like glycolysis itself are then going to be shuttled into respiration stage 3, which is called oxidative phosphorylation. Here we're going to take these electron carriers, and this is the portion that generates the majority of ATP in our catabolic processes. It is going to use movement of electrons to actually drive the synthesis of ATP. So this is just a grand overview. We'll be going into all these stages in detail. Now, I noted that the positioning of these pathways in the cell is important. Where they are localized does lend to how these processes are occurring. Just a reminder that glycolysis, when we talked about it, occurs in the cytoplasm. Gluconeogenesis, when going backwards, remember, occurs almost all in the cytoplasm except for the first step, which occurs in the mitochondria. Now, the other stages of our energy production here are going to occur in the mitochondria. Mitochondria of the cell usually are called the powerhouse of the cell. This nickname comes from the fact that this oxidative phosphorylation mainly occurs within the mitochondria. So the citric acid cycle, all the citric acid cycle is going to occur in the mitochondrial matrix, and that is with the exception of one particular enzyme. Again, we'll talk in more detail about this, which actually is in the inner membrane of the mitochondria. And the oxidative phosphorylation, which is stage 3, that final stage where we're moving electrons, is going to occur in the inner membrane of the mitochondria. So when we're talking about these different membranes, you can see here is a diagram of mitochondria itself. This is the inner portion of the mitochondria. You can see here is the inner mitochondrial membrane. And this is important to note. These portions of inner mitochondrial membrane, which is where the oxidative phosphorylation is occurring, that's inside this whole portion here. And on the outside, not really shown in detail, is the outer mitochondria. So this is the outer mitochondrial matrix where things like the citric acid cycle are occurring. There is a gradient between these two of negative and positive charge, and that is going to be very, very important. This separation and this gradient between the two is how we actually drive these final stages of oxidative phosphorylation. Now let's rewind a little bit. That was an overview of all of the stages of respiration we'll be going through. But for now, let's back up and focus on our good friend pyruvate, where we left off, and look into our first step of respiration. So as a reminder, pyruvate is the product of glycolysis. After our first stages of oxidation of glucose, we can create pyruvate. Pyruvate can be created from other sources, and it has a variety of fates after it is created. So once we make pyruvate, it can, in the cases of animals and some organisms, be fermented into lactate, right? We talked about how our muscle cells will use lactate dehydrogenase to regenerate some NAD plus for glycolysis. So pyruvate can make lactate. In microorganisms, like some yeast and bacteria, it can also be fermented, but it is fermented into ethanol instead of lactate through pyruvate decarboxylase. So these are fermentation pathways that pyruvate can go through. Remember, pyruvate can cycle back and actually be made back into its precursor, PEP, to go through gluconeogenesis, for example. Remember that this step actually is a two-process step in which we make oxaloacetate, and then we regenerate PEP. The first stage of this oxaloacetate is important because oxaloacetate can also be funneled into the citric acid cycle, and we will see it in the citric acid cycle. So this is one potential fate, another potential fate of pyruvate, to make oxaloacetate or to fully cycle back into PEP through gluconeogenesis. Now, the step that we will be focusing on today and the fate of pyruvate is taking pyruvate and turning it into acetyl-CoA. Acetyl-CoA is a molecule that is used extensively in the citric acid cycle, but it is also used and utilized by lipids. It can actually be produced by lipids if we're breaking down lipids. So if we're doing a catabolic process of lipids, we will make acetyl-CoA. Or if we want to build lipids, if there are special lipids in the body that we want to build, in reverse, we can take acetyl-CoA and use it to build lipids. So acetyl-CoA has a role in lipid breakdown and synthesis. But as I said, it is one of the main players in the citric acid cycle through respiration. So this is respiration stage 1. You can see here that the Conversion of pyruvate to acetyl-CoA is as thus, and as written, it looks decently simplistic, right? It's one step. We're taking pyruvate, we're making acetyl-CoA. It seems a bit silly at first to call this a whole stage of respiration when our other stages are all of the citric acid cycle and all of oxidative phosphorylation. But this is a pretty big step. It occurs through a complex enzyme. So this is a protein that's made out of multiple subunits. Remember when we talked about proteins and enzymes that they can have quaternary structure, right? So they can be singular folded proteins that are then interacting with other singular folded proteins into a large complex. So while these have their own chains associated in each subunit, the whole of all of the subunits work together to do one distinct process. Pyruvate dehydrogenase complex is a very, very important complex. And it is made out of 5 coenzymes. These are 5 participating molecules, and there are 3 main subunits that make up the PDC. So we will call this often an abbreviation, the PDC, pyruvate dehydrogenase complex. Again, as I noted, it requires 5 coenzymes. These are small molecules that are involved in the process of this catalyzation of pyruvate to acetyl-CoA. 3 of those coenzymes are what we call prosthetic groups. These are groups that are actually attached to the enzyme, and they are working as a portion of the enzyme. So while they are not a protein themselves, you can see they are molecules, TPP, lipolysine, and FAD. These are all molecules, chemical molecules, but they are attached to the protein and doing a job in the enzyme. NAD plus and this coenzyme A also help in the overall reaction of pyruvate. They are not attached to the enzymes, but they are needed to kind of drive the production of acetyl-CoA from pyruvate. So you can see this process overall is going to be energetically favorable. It does generate some small amount of energy by taking pyruvate, making acetyl-CoA, and note, we also are getting rid of one of our first molecules of CO2 during this whole process. Remember that glucose, when it's breaking down into pyruvate, we do not lose any carbons through CO2, right? Glucose is a 6-carbon molecule, and it makes 2 3-carbon molecules. That's pyruvate, right? Pyruvate is 3 carbons long. You can see here, here, and here. Through this stage, taking pyruvate to acetyl-CoA, we're doing an oxidative step that is going to produce CO2. So we're actually getting rid of a carbon from our molecule. And you can see we're left with only a 2-carbon molecule in acetyl-CoA. So this is one of the first areas where we lose CO2. And take note of which carbon gets lost. It's highlighted here. It's very important because if we were to label these carbons as we went through the process, you would actually see that only this specific carbon, all the way from a glucose, if it was labeled and then made into a pyruvate, would be the one that is lost as CO2. Okay, let's look at the overall reaction of the pyruvate dehydrogenase complex. Here's an overview. This is obviously a very simplified cartoon of each of the portions of the whole complex. These are labeled as E1, E2, and E3, and they go in that order. E1, which is also called the pyruvate dehydrogenase, E1. There's E2, which is dihydrolipotransacetylase, and E3, dihydrolipodehydrogenase. These are the names of each of the subunits. And then these subunits all act together to do the whole process. And you can see within the subunits, there's kind of labeled numbers to tell you each of the stages that occur as one pyruvate is going to become one acetyl-CoA. So in our first enzyme, E1, the first step is to take our pyruvate and decarboxylate it into an aldehyde. This aldehyde is then oxidized to a carboxylic acid. Now, this whole portion is completed through a lipoamide to form a thioester. We'll look again closer at that. This is going to be one of those coenzymes, that are working with the enzyme to help achieve the product. Enzyme 2 is actually where we form acetyl-CoA. So the production of acetyl-CoA occurs right here. So why do we even have the third portion, enzyme 3? Well, in doing this production of acetyl-CoA, we again are going to be using up some of our coenzymes, and they're going to change their structure. So enzyme 3 is there to regenerate the structures that are used in enzyme 2. So the whole reason for enzyme 3 is the reoxidation of the lipoamide cofactor that we previously used, and also regeneration of oxidized FAD. So again, we're going to look at this in more detail and look at each of these stages, but this is the overview of what is happening. Okay, one of the first coenzyme pieces that is acting in this reaction is called TPP. This is thiamine pyrophosphate. Here is its structure. You can see on the left is its structure. Now, this is one of the coenzymes that was brought up. It's actually a form of vitamin B1, which is also called thiamine. That's where the thiamine portion comes from. And it is attached to the first enzyme of our pyruvate dehydrogenase complex, E1. TPP is a common carrier of aldehydes. So our pyruvate is going to actually, in a process, attach to TPP, and it will carry the pyruvate into our second complex. You can see that TPP, as noted, is used in many other enzymes aside from our pyruvate dehydrogenase complex. It's also used in pyruvate decarboxylase, not to confuse you here. Pyruvate decarboxylase is the enzyme that's used in ethanol fermentation. It's used here in pyruvate dehydrogenase to synthesize acetyl-CoA. It's also used in parts of the citric acid cycle in an enzyme called alpha-ketoglutarate dehydrogenase. And it's used in the pentose phosphate pathway. So one of the enzymes that we spoke very briefly about, which is transketolase, also utilizes TPP. So TPP again, has a very important role in many of these enzymes of carrying aldehydes. Note that because it is a derivative of vitamin B1, if you have a vitamin B1 deficiency, it will obviously cause many problems in other areas of the system that rely on this coenzyme TPP. So you see that this thiazolium ring right here in the center of TPP is what is actually doing the work. Here, this very reactive carbon is going to be the site which can bind with a molecule forming this active acetaldehyde. So a aldehyde can come in and bind here to our thiamine pyrophosphate, forming this temporary alcohol, which is then carried through and can be brought into other areas. So just to remind you, what would the structure of this be prior to it binding to the carrier here? We would have a C double bond O H, the CH3. This carbonyl carbon is the reactive portion. That's this carbon right here, which is then going to bind on the thiazolium ring, and in doing so, temporarily makes this an alcohol. So back to our image here, so you can see what's happening in pyruvate. Pyruvate is going to do this process in which it is using TPP to bind and also losing a CO2. So there's a multi-step process here in which we lose this whole CO2. You can see the carbon with the two oxygens. Remember, pyruvate is going to decrease down to a 2-carbon molecule. And in doing this, this carbonyl carbon is going to be the one that attaches to our TPP. That temporarily becomes this alcohol molecule, which is then carried through into our second complex E2. Now, the second complex E2 is going to utilize a coenzyme called lipolysine. This is another prosthetic group. So just like our TPP, it's a molecule, not part of an amino acid or protein. It's a molecule that's attached, bound to the protein, and helps act out the protein's catalysis. Here's what lipolysine looks like. It gets its name from being a lipoic acid plus a lysine residue. So this whole molecule is usually going to be formed where there originally was a lysine amino acid. And the addition of the lipoic acid makes this very, 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 very long chain that we then call lipolysine. And this is covalently attached, actually covalently linked to the enzyme. So it is anchored in there. Now, the lipolysine is made up of this lipoic acid portion, this ring here, which is the area that is going to be doing most of the chemistry. Here it is in its oxidized form. You can see it forms a disulfide, 2 sulfides bound together. These sulfides can be reduced. When they're reduced, they are broken. This is just like what we saw when looking at cysteine. Remember, cysteine can be its reduced form, just the sulfurs, SHs. And in its oxidized form, can form a disulfide bridge. Sulfurs act very commonly in this way, right? They can bridge in an oxidized form and break in a reduced form. In its reduced form, the lipolysine can also act to bond through an acetylated form. So it can bind an alcohol onto its sulfur. There's a reaction in which the alcohol then binds, forming this sulfur here. So going back to look at our cartoon enzyme here, let's focus now on the E2 portion. You can see in E2, here's our lipolysine. It's in its oxidized form, which is nothing has messed with it. It's hanging out, it's oxidized. Now, if we add our aldehyde, or now alcohol that is tethered to our TPP, and we react it with our oxidized lipolysine, this will then bond to one of these sulfurs. You can see where our carbon and oxygen were right here is going to bond onto the sulfur, reforming our carbonyl. So again, it looks just like the end of our pyruvate did prior, but now it's attached directly to this lipolysine. The lipolysine is now reduced, right? Lipolysine is then going to go through a second step. So it now has our piece of the pyruvate, right, attached to it, and it is going to finalize making our acetyl-CoA. This is the second step of E2, forming acetyl-CoA by taking coenzyme A and attaching it to our acetyl group that we formed through this process. Now, this still occurs on our lipolysine. You can actually see in the drawing here, lipolysine kind of rotates fully through using its arm to go through these stages. So it rotates up, it picks up that acetyl group from the TPP, and then it's going to continue rotating. It's going to grab our coenzyme A, make acetyl-CoA, and rotate back through. So remember, in E2 is where we are actually forming our acetyl-CoA. So we'll pick up a coenzyme A, attach it to our acetyl group in step 3, and we have now formed our acetyl-CoA. This whole process is going to dissociate the acetyl group from our lipolysine, but I want you to notice that in the end of all of this, our lipolysine is reduced. So after picking up our acetyl group, reacting it with the CoA group, and making acetyl-CoA the product that we need, the lipolysine is in the reduced form after that process, not the oxidized form. So here's a closer look at coenzyme A. Coenzyme A, we abbreviate it as this CoA, but it is a very, very, very large molecule. You can see it has different portions that come from different synthetic areas or nutrients that we take in. A mercaptoethylamine is a portion that is derived from an amino acid. And then you can see one of the main backbones of coenzyme A is pantothenic acid, which is also vitamin B5. So we're seeing roles for the vitamin Bs that are strongly occurring here. Remember, TPP is coming from vitamin B1, and coenzyme A is coming from vitamin B5. So taking vitamin B5 or coenzyme A itself, if you have deficiencies in these, again, are going to strongly affect your energy production, specifically in making acetyl-CoA. And then at the end here, you can see that we actually have a nucleic acid like molecule with a ribose and adenine. So it has a phosphoadenosine diphosphate portion attached to it. Now, again, we usually just abbreviate it as S-CoA. Coenzyme A, this whole portion of it gets very much just abbreviated into 2 letters. And the portion that is very important is this thiol group at the end. This is actually where it is doing its reaction. So you can see that the acetyl group we picked up from originally from our pyruvate is going to react off of our lipolysine to this thiol group, this very reactive thiol group, which will attach directly to the carbonyl. And that is what makes acetyl-CoA. We've talked a lot about these different biological tethers and how they are acting throughout this complex. So I wanted to just spend a moment talking about their use in many different enzymes. Biological tethers are very important. These long biological coenzymes are important mainly for their flexibility. Notice that TPP, which we talked about, or lipolysine, which we talked about, are both these very, very long molecules. And these types of tethers help us move attached molecules around enzymes to other portions of the enzymes. This allows this multi-complex enzyme to actually do its job faster. If you can just kind of bungee the piece over to the next enzyme complex, then it's going to get over there much faster than if you have to wait on luck that the molecule happens to just drift into that next portion. So here are some biological tethers that we have talked about or you might see in the future. Lipolysine, which of course we just mentioned from our E2 complex of the pyruvate dehydrogenase complex. Also, we mentioned in the past during glycolysis this tether using biotin. So remember pyruvate carboxylase, which is one of the enzymes during, again, glycolysis, uses biotin and the lysine residue tethered together to make this long, flexible tether to help complete its action. Another one that you're going to be seeing is actually a derivative of coenzyme A. It has panthenate and mercaptoethylamine. And this tether, this is a separate tether we'll be seeing in the future, is part of the acyl protein, which is involved in fatty acid synthesis. So again, these long biological tethers, we see different forms of them using different molecules, but you see this kind of common structure of very long tethers with a reactive end on them that are used to move around molecules within a large complex. So the very last enzyme in our pyruvate dehydrogenase complex here that we're going to talk about is the E3 portion. Sorry about that loudness. Sorry. The E3 portion, which as I noted is going to be the portion that regenerates the E2 portion of the pyruvate dehydrogenase complex. So as I mentioned, when we left off in E2, we formed our acetyl-CoA. Yay, we got our product, but that lipolysine was left in its reduced form. And if this protein wants to continue to catalyze and make acetyl-CoA, it needs the oxidized form. because the oxidized form is what is able to react and pick up that acetyl group from the E1 portion. So to reoxidize the lipolysine, we have to have the E3 complex. It is going to be using a actual bound FAD, FADH. So FAD and FADH2, this is an energy carrying molecule similar to NADH and NAD+. It is actually generated later on in other steps, but this particular one is actually bound into the E3. So this is not a free floating FADH2 that can be used for energy generation. It's actually attached, it's acting as a coenzyme in E3. It is what is going to help to regenerate this oxidized lipolysine by taking an FAD and making an FADH2. We're going to reduce the FAD, and by reducing the FAD, we're going to oxidize the lipolysine back to its form. Now, to keep the E3 complex going, that FADH2 now needs to be put back in its oxidized form. And this is done through using a free floating NAD+, which is made into an NADH for this complex. So you can see the overall kind of usage and generation in this process is that we're going to add in a pyruvate. We're going to add in an acetyl-CoA. We're going to add in an NAD+. Oh, excuse me, we're not adding an acetyl-CoA. We're adding in a coenzyme A. Adding in a pyruvate, adding in a coenzyme A, adding in an NAD+. And what we're going to generate is instead going to be our acetyl-CoA product, a CO2, which goes off and is released, and an NADH. So here you can see the realistic view of our pyruvate dehydrogenase complex, now knowing all of its products that it makes. Again, this is why this is a whole full one stage of respiration. It is one complex, and yes, we do technically only take pyruvate and make one product, acetyl-CoA, but there is a lot going on in this whole complex. It's made out of multiple subunits that are even then combined together. So here's the actual crystal structure that came from the PDB here, showing the multi-enzyme complex, the E1, E2, and E3 portions. And you can see that they're actually color-coded, but there's like multiple subunits within this subunit. So there's not just one E1, one E2, and one E3 that are on a whole complex. There's actually multiple of them that are then connected together. You can also see kind of branching between areas. These are those long arms of our TPP or lipoic lysine that are acting to kind of move and branch these pieces to other complexes. So to end here, let's take a moment to talk a little bit about the regulation of pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex obviously is a very important step in energy generation, and thus it has a lot of regulatory molecules. Again, just like glycolysis, we don't want to be investing energy into converting these molecules if we don't need that ATP, right? So there's going to be a lot of regulation around if we're going to use up that NAD plus and make this acetyl CoA. That acetyl CoA has very limited options in what it can be doing. It mainly is going to be just like going into the TCA cycle and generating energy. So if we don't need that acetyl CoA and we don't need to generate energy, or we don't need to make lipids or do things with lipids, then we want to stop the formation of acetyl CoA through this enzyme. So just like glycolysis, we have a lot of allosteric regulators. Remember, allosteric regulators are going to be molecules that are going to bind and reform enzymes in a way that can activate or deactivate them to continue their processing. In the case of pyruvate dehydrogenase, things that activate it are things that showcase that there is low energy. So if there's just regular coenzyme A, not made into the acetyl CoA form, a lot of coenzyme A, that's one of the reactants. A lot of NAD plus, right? This is again one of the reactants. A lot of pyruvate, this is a reactant. These are all going to say, hey, we have a lot of buildup of reactants. Maybe we need to be making products. Maybe we need to make acetyl CoA. ADP, of course, is going to signal that you are low on energy. So this is another way to say, hey, ultimately, we are not forming any ATP down here. There's a lot of ADP that's building up. So let's activate our pyruvate dehydrogenase. And another thing that can activate it is actually calcium. Calcium is a molecule, if you remember, that helps regulate use of muscles. So when you have a large influx of calcium, that is a signal to start using muscles and burning energy. So when calcium influxes occur, this signals that energy is needed. And this is going to occur mostly in muscle cells. But this is one of the activators as well to signal, hey, we need energy. Some molecules that are going to inactivate the pyruvate dehydrogenase are going to be products of it. They're going to say, hey, we have enough products. You don't need to keep making acetyl CoA. So acetyl CoA itself, if there's a lot of buildup of it, will say, okay, time to shut down. You've made a lot of acetyl CoA. NADH, which again is one of the products here, is going to signal. ATP, again, is a signal that we have built up a lot of energy down the line through using acetyl CoA. So we don't need to keep making acetyl CoA to make more energy. And then interesting, one of the other ones that turns it off is if you have a lot of fatty acids around. Remember that fats can be used to actually break down and make energy, or they can be built up using acetyl CoA. So a lot of fatty acids signals that you have high energy reserves, or you've built up enough fatty acids that you need, so you don't need to keep breaking down and making more acetyl CoA. So these are all allosteric regulators. Remember, these are molecules that are going to either activate or deactivate the actual enzyme into working. Another way that the pyruvate dehydrogenase is regulated is through a type of allosteric regulation, but specifically phosphorylation. Phosphorylation, as mentioned, is a very, very common way to turn on and off enzymes. Very simply, there will be a portion of the enzyme that can have a phosphate added to it or removed from it. Usually it's an alcohol group. And depending on if that phosphate is there or not there, it can turn on or off a whole enzyme. This type of regulation is extremely common in many of the pathways that our body utilizes. So, a lot of times, phosphorylation can activate enzymes. In the case of our particular PDC complex, here, pyruvate dehydrogenase complex, phosphorylation actually inactivates it. So adding a phosphate on will turn it off. This is, again, a very, very simple on-off mechanism, right? You add a phosphate, turns it off. You take that phosphate away, turns it on. So whatever state it is in will help it become active or inactive. This taking off or putting on a phosphate actually has to be controlled by other enzymes. So there's a little bit of a bigger picture going on here. So here is our pyruvate dehydrogenase complex. Here's all the stuff it's doing, right? It's on, it doesn't have a phosphate, but let's say we want to turn it off. Well, what is something that phosphorylates enzymes? The name for an enzyme that adds on a phosphate is called a kinase. We've already seen kinases in glycolysis. This is just, there are lots of kinases. The word kinase just means that it phosphorylates. So in this case, the pyruvate dehydrogenase kinase is going to phosphorylate pyruvate dehydrogenase. And when it does that, it inactivates it. These kinases are going to use ATP to do their phosphorylation work. Now, let's say we wanted to go the other way. Let's say we need to turn back on pyruvate dehydrogenase complex, but it's phosphorylated. Well, we have to use another enzyme. As you can see here, this is going to use a phosphatase. We've also seen these in glycolysis. Again, phosphatases, this word is used in many different enzymes, and all it means is that it takes off a phosphate. Kinases and phosphatases are all over their places. They have unique roles in that they will be a kinase for one specific molecule or a phosphatase for one specific molecule, but ultimately they act the same way at their core. This phosphatase is called pyruvate dehydrogenase phosphatase, meaning it's a phosphatase for pyruvate dehydrogenase complex, and it will remove the phosphate. Now, again, like I said, this picture makes it makes it an even bigger picture of what's going on as far as regulation, because the kinase enzyme and the phosphatase enzyme can also be regulated. So their action on whether they are doing their job and turning on or off the pyruvate dehydrogenase complex can also be regulated by similar molecules. So there's a lot of regulation going on. Remember that there's main allosteric regulation. This is the actual direct regulation from these molecules on the pyruvate dehydrogenase complex. And there's regulation by this phosphorylation mechanism that's occurring through kinase and a phosphatase.